right, so for our first of the four categories of tissues, we're going to go with epithelial tissues. This is a pretty big one with a lot of diverse tissues, and it can be a little difficult to tell them apart. So you'll want to take some time with this one to really get to know the tissues, not just on paper, but also visually. So what are we going to do for this particular group of cells and tissue types? We're going to talk about their structure and function as epithelial tissues. We're also going to talk about how they connect together, so different junctions that will connect epithelial cells together. We'll talk about the difference between simple and stratified, as well as squamous or squamous, depending on who you talk to, cuboidal and columnar epithelia as well, and then we'll go into some of the special glands that are usually found with epithelial tissues, like endo and exocrine glands. So let's talk about epithelial tissues in general first. What are some of the things that all epithelial tissues have in common? They generally are large sheets of cells that are going to cover a surface. So this can be a surface that is facing the outside, so like your skin, protecting you from the outside environment, but it can also create linings on the organs inside your body as well. And there's a few reasons why we have epithelial tissues. Number one is protection. So if you think about your skin, we'll just focus on that one since it's the obvious visual that we can all kind of create in our head. If you think about what your skin does on a very basic level, protection-wise, it protects you from all the different things in the environment around you, right? It protects you from physical damage, right? Wear and tear. It protects you against chemical damage, protects you against biological damage. So what do we mean by that? Physical, right? You don't just immediately start bleeding, right, if you bump into something, right? So you have some physical protection against bumps, bruises, heat, cold, right, things like that. Chemical, right? You can have things on your hands, things on your skin without it causing burns, without it causing damage to the tissues below. That if you had, maybe let's say you're using a cleaning product and you don't have gloves on, it's not necessarily going to eat away at your skin and eat away at the tissues underneath. Your skin can kind of absorb some of that damage, absorb some of that wear and tear without causing damage to the more sensitive parts underneath. And then biological, we mean if you think about the environment, it's covered in germs, right? There are germs everywhere, and your skin is actually really your first defense against infection, against getting infected with bacteria or viruses. And this is one of the reasons why, if someone is a burn victim, that the risk of infection is really, really high, because their primary defense against biological agents getting in their body has been damaged so severely. The epithelial tissues are highly cellular, and this is something you're probably thinking, well, is a tissue? Of course it's made of cells. And yes, all tissues are made of cells, but some tissues have acellular components like fibers and whatnot that run through them, and that kind of spreads the cells in the tissue out. But with epithelial tissue, we're talking lots and lots of cells in that space. There's very little material between the cells. So like I mentioned, some tissues have like connective fibers and proteins and whatnot between them. We don't really see that with epithelial tissues. So if you are looking at a sample and you see like a lot of collagen or elastin fibers, then you know you're probably not looking at epithelial, because epithelial tissues don't really have a lot of that. It's mainly just cells densely packed together. And these cells are actually connected through these junctions. We'll talk about the different junctions. These cells do have polarity, meaning we have what we call an apical and a basal surface, kind of like a top and a bottom, right? There's an orientation. They're not just kind of facing in whatever direction. There's a head, there's a tail, right? Kind of end with our cells, and that they're nearly completely avascular. This means we don't see blood vessels running through them. And this is one of the reasons why, if we think about the skin again, you can get scratches on your skin and not see a drop of blood, right? If you get a little scratch or a little prick, or you scrape something, you usually have to go pretty deep before you start seeing blood. So that top lining, the epithelial lining in your skin, doesn't have any blood vessels. So when you scrape it, you don't see any blood because there is no blood there. And that these are connected through cell junctions. We're going to talk about each of those different cell junctions in just a minute. Here we go. So let's talk about the cell junctions first. We have a few different types. We have three main categories: tight, gap, and anchoring junctions. These are not limited to just epithelial tissues. We can see some of these in other locations like the gap junction, but we do definitely see these in all the epithelial tissues. So let's talk about the tight junction first. And as the name implies, it is going to tightly weave cells close together. So we see these transmembrane proteins that almost look like little pearls that have tightly bound these two adjoining cells next to each other. There's very little space in between. And this is fantastic in areas that you need to be kind of like watertight. So you think about like in your stomach or your bladder, like things that are holding like a liquid or holding some sort of fluid, you wouldn't want that to seep through between the cells, right? So a tight junction is going to help create a barrier that is to some degree watertight. We also have the gap junction, which is over onto the right at the top there. The gap junction, if you look at it, looks like little pores that are reaching from one cell to the other. These little pores that are connecting them are called connexomes. And the whole point of this is to kind of create like a bridge between the cells. This is really good for rapid communication. So if you need to communicate with the cells next to you very quickly, gap junctions are fantastic for that. And we'll see them used for that purpose later on in the semester and when you get into physiology. But fantastic because it allows for rapid communication. To communicate between cells without gap junctions requires that whatever chemical you're using to communicate is secreted out into the gap between and then absorbed by the cell next door. And that takes some time. But with the gap junctions, you're eliminating that need to secrete and absorb. You can just communicate straight across. And then the anchoring junctions, this depends on where, what we're trying to do, what we're trying to connect. But the thing that they all have in common is that we are anchoring, we're connecting cells to cells or cells to some sort of baseline or basement membrane, right? That's the big thing with the anchoring junctions is we're trying to connect them so we can't float away from each other. And either we're connecting a cell to another cell or we're connecting a cell to some sort of blanket membrane, basement membrane. So let's look at the two that join cell to cells first, which is the desmosome and the adherens junctions. We're not going to get into the nitty-gritty in terms of what makes these different from each other. That's getting into some physiology stuff right there. Some of the main things are the proteins that we use. So if we make one distinction between them, for us at least, it's that the filament used to connect them, we see that cadherin that connects them, but in the adherens junction we see actin filaments along the inside of the cells, whereas in our desmosome we use keratin. So that's the big difference that we care about right now. I'm not going to go really beyond that for you guys right now, but you should know both of them are used to anchor cells to other cells. And then the hemidesmosome is very similar to the desmosome. The big difference though is instead of connecting one cell to another cell, we're connecting our cell to, in this case, what's called a basal lamina or a basement membrane. So we're not connecting it to a cell, we're connecting it to some extracellular components that kind of serves like a sheet of paper that everything can be kind of glued down onto. Alright, so now let's get into the actual epithelial tissues themselves and talk about how we classify epithelial tissues. And it's kind of beautiful the way that we classify them because it comes down to two things. What shape are the cells, and how many layers of cells do we have? And so you'll always name them starting with the number of layers, ending with the shape. So I know I have it kind of reversed here on the slide, just keep that in mind. So we can have three different shapes of cells when we're looking at epithelial tissues. We can have squamous. I like to think squamous is squashed, so it's flat. Cuboidal, as the name implies, is cube-shaped, so they're about as tall as they are wide. And then columnar, which is like a column, right? So these are going to be cells that are taller than they are wide. They're very tall, but they're narrow. And then the number of cell layers, we have two main types of names for the number of layers, but then we also have a couple special ones. So let's talk about the first two to start with, which is simple and stratified. Simple is as simple as it gets, right? The simplest number of layers you can have is one. So If something is a simple epithelial tissue, there is only one layer of cells across. Stratified means more than one layer. We don't care how many. It could be 2, it could be 100, it could be 1,000. We don't care. Stratified is more than one layer, but we're not making any sort of distinction on how many more. And then there's a couple special ones. There's pseudo-stratified. So pseudo, right? Like pseudoscience. We're talking fake, right? If something's fake, you could use the root or the prefix pseudo. So pseudo-stratified, it appears to have more than one layer, but it actually is only one. So pseudo-stratified kind of fakes you out. It's a single layer that they're not nice and neat, and so it looks like you kind of really have to pay attention because it kind of looks like it has more than one layer. And then transitional, we're going to change our shapes. We are not necessarily squamous, cuboidal, or columnar only. We have a changing in how these cells are structured shape-wise. And this is great in areas that need to be able to stretch or move. So like we see this in the bladder because the bladder has to go from something very small to be able to stretch to accommodate the collection of urine. Otherwise, you would have to urinate all the time, right? So that transitional epithelial tissue in the bladder allows you to hold it a bit longer, so you're not having to urinate every hour or so. All right, so let's take a peek at the visual for each of these. So let's look at simple ones first, and then we'll look at stratified. So we'll go down kind of the lines here. So simple, again, if you notice, every single one of these down this column has a single layer, right? There's our basement membrane, or the basal lamina, as it is called, that they're anchored to. So we always have that basement membrane, that basal lamina that we're going to be anchored to. And you notice that every single cell is anchored to that basal lamina, right? That's an important thing, right? That's how we are one single layer. No matter how flat or tall we are, right? Everybody's anchored down to that basement membrane. The squamous ones are flat, right? They look like little fried eggs that are just kind of plopped on there. Cuboidal, again, we see it's not perfectly square, but it is about as tall as they are wide. And then the columnar ones we see, again, very tall compared to their width, right? They're very narrow for width. And so all of these are simple. You'll notice in the name, we put simple at the start, and then we name them after the shape. So simple squamous, simple, one layer, squamous, flat. Right? Simple cuboidal, one layer, cube-shaped. Simple columnar, one layer, tall. On the stratified side, we see multiple layers. And again, we see like 2 layers here, and here we see, was it 1, 2, 3, 4, 5. So we don't care how many. We just care that there is more than one. So stratified squamous, we see lots of them stacked up, and we see that there's one layer that is anchored to that basal lamina, but the rest of them are anchored to each other. They're not anchored down all the way to the bottom. Same thing with stratified cuboidal and stratified columnar. The pseudostratified, again, if you look at it, and it's maybe a little bit more obvious in this picture, but when you're looking under the microscope, it can be more difficult. This one's a little tricky. If you're looking at it without looking at that basement membrane, you might assume that this cell here is one layer and this cell here is a layer above it. But if we pay attention, we see that all these cells, right, all 8 of them in this picture, all are in contact with that basal lamina. That's why we call it pseudo-stratified, because it looks like, if you're not paying attention, you might say that it has more than one layer, but if you really pay attention, you notice that they really are all in contact with that basal lamina, so they have to be one layer, even though they're trying to fake you out. So let's take a peek now at some of the actual images of these cells. And this is one thing, again, the epithelial ones can be a little confusing, simply because they all tend to have a similar look to them to some degree. You're going to see a lot of pinks and purples, right? So let's go ahead and take a peek. So let's look on the left first, where we see these flat looking cells, like we're looking from the top down. We see that there's this dark line around them. That's the cell membrane, and then it almost looks like a little egg with a yolk, right? That yolk looking structure is the nucleus inside each cell. And then the lighter color around it is the cytoplasm. All right, so these have been stained so that the nucleus stands out, the membrane stands out, but the internal component, the rest of the organelles don't really stand out, and that's okay. If we look at the simple squamous cells in the picture on the right, right? We're looking at them from the side, and so we see like there's a single flat cell next to another single flat cell all in this line going all the way around here. That's our simple squamous. Keep in mind, and this is really important for all of the histology slides we look at, all of the histology stuff we do in class, most of the slides we look at will have more than one tissue type on them. So you really do need to pay attention to which part of the slide the label is referring to, which part of the slide is actually what you're supposed to see, because it's just physically impossible in some cases to truly tease apart the tissues from each other, right? So in this case, we see some other ones, right? We potentially see other types of cells, but we do see the simple squamous right here. So we just want to make sure we're paying attention to that. Simple cuboidal here, right? Again, we see about as tall as they are wide in a single line, right? If we look around here, bam. And you'll see a lot of simple cuboidals in like glands. So when we look at certain like tubules within like the kidneys and whatnot, you'll see simple cuboidal. Quickly going back before I forget to the simple squamous, where do we see that? We can see simple squamous in places like the lining of the lungs. So the very inner lining in the lungs, the very inner like alveoli and whatnot, could have simple squamous found in them. And then simple cuboidal, like I mentioned, we're thinking things like glandular things. So we'll see these in like inside like tubules within like the kidneys. And then simple columnar epithelium, again, we see against one single layer, right? One single layer, and they are taller than they are wide. So like we see that they're rectangular in shape. It's probably a better way to put it than tall versus wide. So the narrow part is in connection to that membrane, that basal lamina, and then they extend far out from that basal lamina. Simple columnar is probably the easier of these three to identify, simply because the shape gives it away a little bit better than the others. But we do have some pseudostratified columnar epithelium, so this would be something taken from like the trachea, and so we see that there's a single line of cells, although they are kind of wrapping around each other, kind of giving each other hugs and looking a little skinny versus a little wider, and so this is all connected though to this basement membrane, right? So that's why we call it pseudostratified. It looks like it might be more than one, but in reality everyone's connected to that baseline. In this case, we do see cilia as well, so we could say this is a ciliated pseudostratified columnar epithelium if we want to add more into the name. And this would just be something like, if we're talking about the trachea, that's used to sweep mucus around. What we see too, especially if we're looking at the trachea, along with the pseudostratified columnar epithelium, and kind of the reason why it's pseudostratified, why they don't all mesh up so evenly, is because of these goblet cells that are hanging out in between them. So if we're looking at the trachea and what the trachea does, besides provide an airway down to the lungs, it also has these little cilia and these little extensions to help sweep debris and mucus out. But where is that mucus coming from? The mucus is coming from these goblet cells, right? So we see secretory vesicles with mucin in them. So these are the ones that are producing the mucus to help protect your lungs from things getting deeper in, right? So when you breathe air in, it's not perfectly sterile, it's not perfectly clean or filtered, and so things like the hair, the cilia, things like the mucus can trap that and move it up and away from the more sensitive, deeper parts of your respiratory system. Now let's go to stratified epithelial tissues, and so we're going to see some examples of each. So the best example of stratified epithelial tissue is your skin. So we see this sample here. We see stratified squamous, right? Stratified squamous, where we see lots of flat cells all stacked and stacked and stacked in lots of layers, right? And again, remember, stratified just means more than one layer. We don't care how many more. Here we see those stratified cuboidal, right? More than one stacked on top of each other, just about as tall as they are wide. And then over here we see our stratified columnar, where again they have some height to them, and so we have two layers in this case, but they are taller than they are wide, right? That's the big thing there. And then transitional epithelium, I know this picture is a little blurry, sorry about that, it's the best one I had, but we don't see a single shape, right? And so like in the bladder, this is where we could find that. This would be fantastic for stretching and changing shape, right? For areas that need to stretch and change shape. So like your bladder, for example, you need to hold urine. If your bladder did not have the ability to change shape and stretch, then you would not be able to hold much. And so you'd have to urinate very often. Also, the stretching is what is used to tell your body that your bladder's full in the first place. So having this ability to stretch is a twofer, right? It helps you hold more urine, but it also gives your body a way of knowing when the bladder is full, when the tank is full. All right, so now let's talk about some of the glands that we can find in the epithelial tissue, so glandular epithelium. We can have endocrine and exocrine glands. The big difference between the two is where things get secreted. Both of them are going to cause secretions to occur, but what those secretions get into and where they end up is the difference between endocrine and exocrine. So endocrine, this is where we are sending secretions directly into the surrounding tissues and fluids. So a lot of times we think of endocrine glands as things like organs or glands that release into the bloodstream. Exocrine glands use a duct though. So they have some sort of duct that opens directly or indirectly to some sort of external environment. They can be unicellular like the goblet cells, or they can be multicellular. So the goblet cells we saw that secrete mucus, they're secreting mucus into the trachea, like the open space in your trachea. Those would be exocrine glands that are unicellular. So let's take a peek at some exocrine glands, because there's the goblet cells that we looked at, but there's also a couple others. So let's look at some. So the alveolar or the acinar are going to be some simple ones. So things like the simple alveolar one. We don't see these in adults, so we won't really talk about them beyond right now. But we do see things like the branched alveolar, the simple branch, where we are looking at sebaceous glands, like in your skin. So the glands in your skin that add oil, that help lubricate and moisturize your skin, are going to be simple branched alveolar exocrine glands. We also have simple tubular, right, glands. These are going to be what we find in like our intestines. We have a simple coiled tubular, so merocrine sweat glands. We have different types of sweat glands. This is one of them. Simple branch tubular, so we find this in like the gastric glands, like in your stomach, and the mucus glands that are in your esophagus. We also find them in your tongue, so you think saliva, and then your duodenum, which is going to be that first part after your stomach, so the first part of your small intestines right after your stomach. When we get to the compound ones, if you notice the difference, right, simple has just one kind of piece, right? You're either simple, you have one thing coming down with a couple branches, right, or you're curling around, you're straight down. But the compound ones, you see more than one, right? So like more than one of those like clover shapes, more than one of those like little duck feet or bird feet shapes coming down here on the compound side. So let's look at compound alveolar first. So this would be like mammary glands to produce things like milk. The compound tubulo alveolar. So we have the tubes and we have the little clover leaves. This would be things like in your salivary glands, as well as glands in your respiratory system, in your pancreas. And then the compound tubular mucus glands, like in your mouth. The bulbourethral glands, when we look at men, are male reproductive systems, as well as in the testes. So those ones, the best examples we have are mainly in the male reproductive system. But this is something to take a peek at. You should at least have a baseline understanding of what an exocrine gland is, no matter what, and you definitely should know the difference between simple and compound. Most of the other parts of the names are just giving away the shape. The only part that's not very obvious would be the alveolar part. So how can we secrete things? There's a few different ways of secretion. We have merocrine secretion, apocrine secretion, and holocrine secretion. So all this comes down to is how the secretions are released when it comes to determining merocrine, apocrine, and holocrine secretion. And so we'll go into that in just on the next slide, because there's a picture and I'm not going to try to describe it very well. And then we also have types. So what type of secretion are we releasing? So sebaceous glands, these would be, or sebaceous secretions would be watery, that have a lot of enzymes in them, versus mucus ones, it's going to be watery but also could be very viscous because we have mucin in them. So we think kind of the difference between them as consistency. So let's talk about the modes of secretion though. So it really just comes down to how do we release these things. So American secretion, right, this is where we have all these little vesicles containing the secretions we want to release, and those vesicles fuse with the outside and release the contents. One example, we have American secretion, sweat glands, right, that can just release the sweat this way. So we're just creating a bunch of vesicles and then release them. Apocrine secretion is a little bit different because rather than release the contents of the vesicles only, we're actually going to pinch off a piece of the cell, right? So, and this is also another type of sweat gland you have, are apocrine sweat glands, but we actually pinch off a piece of the cell, right, in the entire thing, in our secretion. Holocrine is probably the most metal of the three because we're actually going to require that the cell dies to become the secretory product. So Merocrine is kind of like the simplest one. It's probably what you would think of when you think of a cell creating a secretion, and that the secretion is made, it's in a vesicle, it spills outside the cell just keeps doing its thing, on and on and on. Apocrine kind of makes sense. You might have thought like, oh yeah, that could happen, where you pinch off a piece of the cell itself to become that, but holocrine is really the one that's probably a bit extreme, in that the cell is actually having to die in order to cause that secretion.